Welcome to Outsmart Your Genes, where science-based strategies help you shape your genetic destiny for a healthier, longer life. I am Dr. Lucia Aronica, lecturer in epigenetics and nutrigenomics at Stanford University. Today's episode might sound like science fiction, but it's not. Right now, in a lab at Stanford University, scientists are working on something like a molecular time machine for our cells. And the person working on this revolutionary technology is our guest today, Dr. Vittorio Sebastiano, Associate Professor at Stanford School of Medicine, co-founder of Turn Biotechnologies, and pioneer of the groundbreaking ERA technology, or Epigenetic Reprogramming of Aging. Now, when you think about women's ovaries and aging, you probably think it's just about fertility, right? But here's what's fascinating. These organs are more like conductors of an hormonal orchestra, sending signals that affect how our entire body ages. And today, we will explore how Dr. Sebastiano's ERA technology might help reset this aging button. But today's episode isn't just about women's health. Dr. Sebastiano will tell us how HERA might rejuvenate everything from our skin to our immune system. And stick around until the end because we will have a fertility lightning round where Dr. Sebastiano will share his opinion on today's most popular fertility supplements and interventions. And as usual, if you are interested in science-based content on health and longevity, make sure to subscribe to the channel. And now let's welcome our guest, Dr. Vittorio Sebastiano. Vittorio, thank you very much for being with us today.
Ciao Lucia, buongiorno. Thank you for having me. It's a great pleasure and a great honor.
Yes. You know, some of the best collaborations start over a meal. And that's exactly how we met years ago. It was at the Stanford Lichashine Dining Hall. And what started as two Italians sharing lunch ended up in an incredible journey. We ended up co-teaching Stanford's first professional program in epigenetics, which has sparked so much interest in the personalized medicine and longevity medicine space. So now we have a lot to catch up today. And I would like to start with a provocative question. Does aging have a reset button? And if it does, where is it located in the cell and how does it work?
The answer is that absolutely yes, aging has a reset button. It's a little bit complex, so I need a couple of minutes to unfold why I strongly believe.
Yes, please. The mission of this channel is making epigenetics mainstream. So feel free to simplify. Sometimes, simplifications end up not to be too accurate, but I think the most important thing is that people understand.
Right. So, the reason why I believe that aging has a reset button and that this button can actually be engaged and activated, and so aging can be reset, is in the fact that I strongly believe, and I'm in good company actually. I'm not the only one thinking so. I strongly believe that the fundamental linchpin or the fundamental explanation of why cells, and obviously as a consequence of that, tissues, organs, and the entire body age is because there is a progressive loss of information in the so-called epigenetic program. I'm sure that your audience is already familiar with this concept, but let's just very quickly recap what it is. The analogy that I always use to explain to a lay audience what the epigenetic program is the analogy of a language. Let's take Italian just for the sake, since we're both Italians.
Yeah.
Let's take Italian as an example. In the Italian language, there is a basic layer of information, which is the alphabet, right? We have 21 letters that we currently, typically use in our alphabets, right? Now, the letters on their own are meaningless, right? So in order to build a language, you need to assemble those letters in words, right? And then those words need to be assembled in a meaningful fashion in sentences, right? How do you do that? Well, you use two set of rules, which are the grammar and the syntax, right? To kind of take words and use them and combine them in a meaningful fashion so that now you have a sentence that makes sense. Okay, so this is the starting point. The cells behave in a very similar way, in a similar fashion. The alphabet, of course, is not comprised of 21 letters in the case of the cells, but it's only 4 letters, A, T, C, and G. These letters can be combined and assembled in our genome in different ways, in different stretches, in different combinations, which are the words that in this analogy are the genes in the cell. But the question is, well, how is it possible that the same information, meaning the same words, are used so differently in different tissues and in different cells, so that, for example, a neuron is a neuron and is not a liver cell? Well, that's because of the epigenetic program, which is the syntax and the grammar that the cells utilize to turn on or use only specific set of genes, so that in neurons, you have a specific set of genes that is neuronal specific. And in the liver, you have other specific set of genes that are liver specifics. So this is the analogy to explain, hopefully, in a clear fashion, how this language and this information work in the cells. Now, what happens with age? Well, what happens with age is that because of, well, for sure, a developmental program which is embedded in our genes, but also because of the exposome, meaning we are constantly exposed to damage, to inflammation, sun exposure, our lifestyle, of course, diet, pollution. These exposome, which is a weird word to say, the exposure to environment, has an impact on the epigenetic program, because the epigenetic program responds in real time to signals from the outside, because it needs to adapt very quickly to the environment. And so what I'm trying to say here is that because of these insults that are coming from the outside, the epigenetic program progressively becomes dysfunctional. And because of that, for example, to go back to the example that we made before, neuronal cells at some point with age start activating genes that are not supposed to be active, or silence genes that are not supposed to be silenced. And the same is true for every tissue. So to make a long story short, the epigenetic program is the fundamental information that dictates how cells should behave. With time, this information gets lost. But the good news is that the epigenetic program can be reset, can be reprogrammed. And so we can potentially bring it back to a youthful functional state and reinitiate a youthful program of cellular function.
That's a great analogy. I use different analogies, but I like this one. So it's like a language, it's made of syntax and rules. And as we age, also our cells age and start to make mistakes in the language. And so the next question is and how we are going to reset this aging button, and what is this epigenetic reprogramming of aging technology all about?
Right. So before explaining what ERA is all about, let me tell you, and we go back to, I think, that the theme of today's conversation, why the ovary and why the egg, and why it's so.
Okay, let's
Women
start with that, because it was my next question, but let's start. I think that's important because people are here for this reason. So
Right.
let's go back and ask, why ovaries are so central to aging? Not only isn't just about fertility, right?
Right. So I call it my epiphany, the epiphany that I had about 11 years ago when I started thinking about the problem of aging. And the epiphany obviously was there's always a reason why you start thinking a certain way, right? And the reason why I started thinking about aging from an ovarian centric, or as I call it, the egocentric point of view,
I like it.
is that I really realized, thanks to the fact that I am a developmental biologist by training, I studied development and reproduction my whole life. So this is my passion. I came to the realization that we have been looking for the fountain of youth, or really the way to rejuvenate cells, for forever, right? We as human beings, we're obsessed in a way with this quest. Well, when I started thinking about this problem, I said, well, wait a minute, but nature has already figured out a way to not age. And it's just a matter of understanding how it works and apply it to other cells. What do I mean by that? If you think about the fact that every time, and this is true for every species, not just for us, every time we have babies, right? The babies are not born with the chronological or the biological age of parents, right? They're always born age 0, right? And this is just fascinating, right? How is it possible? Because even the germ cells, meaning the cells that are responsible for creating a new organism, age. So how is it possible that this glimpse of age is not vertically transmitted to the following generation? And that the reason is because the egg is a phenomenal machinery that can actually rewire, reset the epigenetic program of the germ cells and bring it back to a pristine age 0 state that allows then for the new life to continue with the expected lifespan and health span, which is obviously then dictated by behavior, right?
Yeah, and this is an example. Just I want how how nature actually resets this aging button at every generation. So it is what there is an inbuilt system doing that, right?
Exactly, and it's the most perfect inbuilt system that exists. And but the point is that nature, and nature couldn't care less about the fact that we as individuals, right, age. The most important thing is that life continues, right? And so if you do an intellectual experiment and you say, oh, okay, so this is true between myself and my parents, right? I was born age 0. The same is true for my parents and their grandparents, and then the grandparents. And so if you go back in time, you realize that at the cellular level, life has not aged over the course of 3.6 billion years of evolution on this planet, meaning that cellularly we are connected to the first form of life that appeared on the planet, again, 3.6 billion years ago. So that's already, I think it's really an incredible realization, and that was the epiphany, to say, well, at the cellular level, this already exists. But so can we learn how that process works? And can we apply it now to other cells in the body that, of course, age and age very rapidly with time? Meaning, can we apply the same principles to the skin cells, to the brain cells, to the muscle cells? So the clue here, or the key here aspect here, is really to understand how that process of resetting this epigenetic program works, and apply the same principles or similar principles to other cell types. And that's exactly how ERA came about, because ERA is inspired by these principles of reproduction, if you wish, and is a way to apply those principles of reproduction in a simplified fashion. Of course, there's still a lot that we need to learn and understand, but in a simplified fashion, to other cells in the body.
Yeah, so the ovaries are really like an example of how nature has figured out already this resetting process. And also they are like the conductors of an hormonal orchestra that's sending signals that really affect how we age systemically. It's not only about fertility, and women know this very well, how hormones rule our life. So yeah, so maybe then you can tell us more about ERA now. What
Sure.
do you think?
Yeah. So ERA basically is as I said, is a simplified version of this idea of resetting the epigenetic landscape. And the way it works is very simple. So what we do is to bring inside the cells a cocktail of mRNAs, of messenger RNAs. Messenger RNAs, probably, I mean, you're familiar with it. So these are molecules that can encode for proteins. Once are inside the cells, the mRNAs encode for the proteins, and the proteins go into the nucleus of the cell and reset, because they know how to do, we're using special factors that know how to do these things. They go into the nucleus and they reset this epigenetic information that I mentioned before. And so in a way, they clean up the mistakes that have been accumulated over time, and they bring that program back to a youthful state. As a function of that, the cells now have the epigenetic program that is now youthful and functional, and as a consequence of it, they're not just molecularly youthful, meaning that they express the genes of youthfulness, or they are epigenetically, you can use clocks, for example, to measure the age of the cells. The clocks we have shown to go back to a youthful state, but most importantly, functionally, the cells are youthful. And so that's right. In principle, ERA can be applied to any cell type, and we are now planning, and we have already started using this technology also on the ovary, because I think that the rejuvenation of the ovary is going to be key for a number of other reasons. Because the ovary, in addition to being the place where the egg, so this special machine of reproduction, is located, the ovary is also central for the process of aging in women. And so tackling that organ, making sure that maybe we can rejuvenate it or bring it back to a functional state, could have potential implications for women's health span.
That's okay. So before I ask you additional questions on the ovaries, let me unpack this, what you just said. So basically, we have figured out the secret agents in the cell, the proteins that know where this reset button is located at the epigenetic level. And this discovery also stems from a discovery that was awarded the Nobel Prize in 2016 for the so-called Yamanaka factors that reset, right, the epigenetic software or language to reprogram an adult cell to a pluripotent stem cell, which is younger. So we know these factors, and now you have figured out a modified technology with modified proteins. And what you do is actually you inject not the protein themselves, but the mRNAs are going to produce those proteins in the cells, right? Why are you working with mRNAs and not the protein cocktail itself?
No, you're absolutely right. The part of the inspiration of the work came absolutely from the seminal work of Shinya Yamanaka that got the Nobel Prize. Yes, and we are using an optimized cocktail that in part includes also the Yamanaka, the so-called Yamanaka factors, plus two more. And what we are doing is in a way similar, but also very different from what he has done. Yamanaka has shown that if you express these proteins for weeks, you can turn, let's say, a skin cell into an embryonic-like cell. What was not realized was that by doing that, you're not only making, let's say, a fully differentiated skin cell into a pluripotent cell, meaning capable then of giving rise to any cell in the body. What's happening also here is that you're making an old cell into a very youthful, almost age 0 cell. So our hypothesis was, okay, if this is true, maybe if we can reprogram the cells shortly, very shortly, maybe we can not turn that cell into an embryonic cell, because that could potentially have issues then, because then you have to redifferentiate that cell back into a skin cell, for example. We don't want to do that. We want to keep the skin cell a skin cell, but maybe we could rejuvenate it, right? Because maybe during this process, we can start erasing those mistakes that have been accumulated in the cell. And that's exactly what we have shown. If we can reprogram, meaning express those factors very shortly, in dosage and in duration, we can achieve rejuvenation without achieving a cell identity loss, meaning that we're not making an embryonic cell. And this potentially could be revolutionary, because if you know how long you reprogram the cells, you can potentially go directly in vivo. If you have an aged tissue, you can shortly reprogram that tissue and you make a younger version of that tissue, but it's still the same tissue. It's just a youthful version of itself.
That's a great explanation. So basically, you have figured out a way to modify the original technology to basically have a molecular time machine, but with a precise timer, where you can stop the resetting process by using mRNAs, because if you inject directly the protein, then you don't have a control. But because the mRNAs need to be translated in the protein, you can stop that process. And by controlling that, you can have a timer and say, okay, don't reset any further. I want to rejuvenate your eggs. I don't want them to turn into pluripotent stem cells, right?
Exactly.
So yeah, that's fascinating. So now back to ovaries. You were talking about ovaries, and I've listened to some of your talks, and where you explain how mice without ovaries live a shorter life. So why is this the case?
Yeah, this is not true just for mice or for animal models. I think it's true also for human beings, for women. There is a clear connection between the loss of function of ovarian activity and the process of aging, which means morbidity and mortality associated with that. Let me explain what I mean. Let's start with the mice first. In mice, there is a lot of literature showing that if you take a young mouse and the term is ovariectomize it, meaning you remove surgically the ovaries, so the ovaries are no longer there. The mice age faster, and they have reduced lifespan and reduced health span at the cognitive level, of course, bone density, muscular function, and so on and so forth. So this is a clear indication that the absence of an ovary in a young individual brings to premature aging and brings to a short lifespan. On the contrary, it has been shown that if you take an old mouse now, you remove the old ovaries and you transplant in the mouse young ovaries. Now these mice live, compared to the controls where you don't do this thing, they live longer and they have a better health span, meaning that their cognitive function is improved, the bone density is improved. Overall, they just look younger, and they live longer. The same
Which
is
is, I
true.
think, significant, considering that anyway a surgery, you might think that a surgery will negatively impact lifespan, a surgery like that, right? And the fact that it doesn't, that actually prolongs it, I think it's very significant.
Right. And the same is true for women too. So let's start with the non-physiological menopause. What I mean by that, there's many women that when they are in their late 20s or early 30s have two conditions that are called premature ovarian failure or premature ovarian insufficiency. So that means that for some reasons, some of which are known, some of which are not known, the ovaries shut down. They stop working. They stop secreting, hormones, and these women enter menopause prematurely, but which means that they also enter an accelerated process of aging prematurely. And that's one of the most extreme cases of this. There is a lot of other women, for example, that have, because of cancer, need to get the ovaries removed. So that's a surgical kind of situation where the ovaries are no longer there. And so for the body, it's like the ovaries are not functioning, right? So they also enter a premature phase of aging. And then there is also data suggesting in physiological menopause, typically women enter in menopause around age 50, more or less. And there are some studies showing that the more for every year that you add, so for every year of menopausal age, you can gain an adjusted rate in decrease in mortality and morbidity of 2%. So that means that if you gain 1 year, you have 2% of chances to live longer and to live better, which is pretty fascinating. So what I'm trying to say here, that there is a clear connection between ovarian function and aging. And so I think it's about time to start thinking about the idea that maybe if we can preserve that ovarian activity, not for the sake of fertility, but just for the sake of making sure that the ovary functions properly in the woman's body, maybe we can start addressing these problems. And of course, this would have a tremendous impact for those women that actually have POI or POF, as I mentioned before, because that's a life-changing opportunity. But potentially we could affect the pretty much every woman, during the physiological menopausal age.
Very interesting. You mentioned we don't know all the factors involved in premature ovarian aging. Among those, the factors that we know are at play, what are the ones we can control, and do they apply also? Can they influence also the menopause in normal people, like when we start menopause, and the ovarian aging for everybody in general?
That's a very good question, Lucia. And the answer is that, well, for the premature ovarian failure, I guess for now, what we know is primarily, we know that there are some genetic reasons why this is happening. For example, there is a syndrome that is called Fragile X. It's a genetic condition where there is a specific change, there is a specific mutation in one of the genes of the X chromosome, and because of that change, the ovary shuts down prematurely. In the other cases, we have no idea. We call them syndromic situations, where it's happening, but you don't know what is causing it. And of course, it could also be related to other factors like I don't know, pollution, microplastics. Who knows? The real answer to your question is that despite this research being so exciting, so fascinating, so important, we haven't done our job over the course of the years, because unfortunately, women and female in general, even in the animal models, have never been at the center of the attention of the scientific community for a number of reasons. One of which being, of course, that because of hormonal cycles, because of changes in the physiology of the body during your hormonal cycles and so on, the studies on drugs and have been primarily focused on men, which are actually more stable physiologically over time. And if you look at the literature, and now there is actually a lot of, we're revamping this idea. There is a lot of interest from the NIH and from the FDA, to really push a lot of attention on women's biology, because most of the drugs that are out there have been tested on men and have been tested on male animal models. And we're realizing that actually, in women, they might have some effects that were not anticipated because they were just not studied in women. I think it's a shame. I think it's just unacceptable. And I think it's about time we start changing the rules of the game.
Yeah, that's a very good point. My guess is that many factors are at play in terms of environmental, the exposome factors you were talking about. It's for sure, as many things in health, a combination of nutrition, activity, and emotions, but also toxicants. Actually, I wanted to ask you a question I received from some followers. So, microplastics. So I know that some research shows that microplastics are everywhere, virtually in every single organ in our body, including ovaries. I think there is almost a teaspoon of microplastic in our brain. And these microplastics can work at the molecular level as endocrine disruptors, so disrupt hormonal balances. So I wanted to ask you first whether you think this might be a factor, and second, I've seen some of your experiments demonstrated that ERA can help counteract the effect of some toxicants for chemicals that are toxic to eggs. Could that effect be transferred also to things like microplastics or other toxicants that might contribute to ovarian aging?
Well, to answer your first question, I think that this is something that we really need to study thoroughly. It's not an easy task because we need to do some longitudinal studies and make sure that yeah, you're absolutely right. Microplastics are everywhere. They're everywhere in our bodies. They're everywhere in the food that we eat, in the water that we drink. We drink water most of the times from plastic bottles, right? And we ingest an incredible amount of pollutants. What are these doing to us? We have no clue, and we just need to really understand what it means. To answer your second question, I mean, I don't want to. I'm very down to earth. I don't want to give false hopes. I don't want to say that ERA is the universal solution to all of the problems. For sure, there is a reason to believe that yes, it could. You mentioned some studies that we are doing. We have induced. There is ways to induce damage to the oocytes using some drugs in mice, of course. We have used those drugs, and those drugs, can selectively kill the eggs, the germ cells in the ovary. And we have seen that if we do that, and also in combination give ERA to the ovaries, the oocytes seem to not die. And it's exciting. It's very early work. Obviously, we need to understand why this is happening, and because that's the most exciting and important reason. So yes, there is potential, absolutely. So like there is potential in many other interventions that are already available out there, or they're being studied. Senolytics, metformin, rapamycin. There is an exciting phase 1 clinical trial on rapamycin from a colleague of mine. She's phenomenal. She's working at Columbia. She's studying rapamycin and its effect on ovarian aging, and she's already in clinical trial. So there is
Wow,
a lot
that's
of
interesting. I would love to interview her for a next episode. Maybe we can talk about it. And in the meantime, while these technologies are available, I just want to suggest our listeners we can't avoid exposures to microplastic completely, but we can limit it. I think one of the best investments you can do is a reverse osmosis filter in your home. It costs like $200-300, but then you save money on plastic bottles forever. And I think I read a statistic where there is 500,000 microparticles of plastic in for each liter of plastic, of water in plastic bottles. So I think this is one intervention. And then the second is we can't eliminate the use of plastics, but at least we can use it with following some rules that minimize the leaching of monomers, of little particles into the food. Like, for example, if you need to carry water, first of all, fill your own water, osmosis water, and then keep it cool and drink it, quickly. And then, so is what is cold, the cold, then avoid heat, avoid acidic foods. For example, don't put lemon in your water in the plastic bottle, or avoid canned. Also, the canned food contains microplastic, so especially the acidic ones, canned tomatoes. We use a lot of them in Italy. Don't do that. Go for glass, and oil. Also, oil increases the leaching of monomers. So if you really need to bring something in a plastic container, don't bring oily stuff. And I think this is what I do. I don't panic, but I apply some rational rules, and I feel I'm doing something. I'm sure I still have a lot of microplastic in my body, but I hope is at least less than the general population. So, Vittorio, I wanted to ask you something, something that was counterintuitive for me is that mice. So when then you remove the eggs in the transplanted mice, the mice were transplanted with young ovaries, I think mice live longer, right? Which was, I was like, why? Do you know why?
I don't know why, and this is something that actually would be very interesting to follow up with. This has been shown in a couple of papers, not by my group. So
Okay.
it's literature showing that if you do the same experiment that I mentioned before, so you take an old mouse, you remove the ovary, and you transplant young ovaries. If you transplant young ovaries, but before doing that, you remove the oocytes from the ovary, the mice seem to be living longer than if they were transplanted with oocytes containing young ovaries, which is counterintuitive. I have to be honest with you, because there is a lot of consensus about the idea that maybe it's the number of follicles, or meaning the number of oocytes in the ovary, that dictates the life of the ovary. So it's very interesting, counterintuitive, and surprising. So yeah, we need to follow up with those studies and understand why maybe this is happening. It could also be that maybe it's the combination of the fact that you have a young ovary in an old mouse that makes the trick. So maybe generally speaking is not true, but if you do that specific experiment, maybe it's true. So that's a possibility. But again, another reason to keep working on this.
Yeah, biology is complex and so is research.
Yeah.
Now, switching gears to other organs, right? So what do you think, what are the organs that affect aging most after ovaries? Also for men, right?
Yeah. Well, my next favorite organ is the thymus, which is also an organ that we're actively working on. Why the thymus? So the thymus is an organ that is located, approximately here in your chest, just above the heart. After the ovary, I believe, is the first organ that ages the fastest in the body. It starts about aging at around puberty, more or less. And by age 50, 90% of the thymus is completely gone and replaced by fat tissue. Now, probably most of you haven't even heard about the thymus. So why the thymus is so important? The thymus is important because it's the organ where most of the T cells, the immune cells that are responsible for killing pathogens and for recognizing our own body as safe, get instructed actually to do that. What do I mean? So in the thymus, the T cells receive the information to be able to kill what's non-self, for example, viruses, bacteria, pathogens, and so on and so forth. But at the same time, they receive the information to not kill and not attack our own cells, because otherwise that would be super dangerous. What happens with autoimmune diseases? It happens that the T cells lose that information, and all of a sudden they start thinking that our own cells are bad, are pathogens, and they start killing them. The autoimmune diseases are actually caused by that. And so the thymus is important because that's exactly where they get instructed to work properly.
And this is why also we need, because we have an increased rate of autoimmune diseases, or also little autoimmune reactions or allergies with aging. Is this the reason why?
Yeah, absolutely. It could definitely be the reason why. So with 90% of the thymus gone, so 90% of that tissue not being able to instruct the T cells, the T cells no longer know or no longer get instructed actually to work properly. And so this is one of the reasons why we undergo the so-called immune senescence, which means aging of the immune system. Obviously, there's much more going on, but this is one of the things happening. And yes, it's absolutely possible that because of this, we are more susceptible to autoimmune diseases, more susceptible to allergies, more susceptible to pathogens, more susceptible to cancer, because we don't know how to recognize and contrast, these bad guys in the body. And on the flip side, we start killing our own cells because, again, we don't know, the T cells no longer know how to handle with that information.
Yeah. You mentioned cancer. So actually, we produce, I don't know how many, I don't know if you know how many, but lots of cancer cells every day. And the reason why we don't develop cancer until we do is because our immune system is actively killing, recognizing those cells. So yes, even immunotherapy is one of the most promising interventions for cancer, and immune aging is one of the reasons why we have more cancer. So on one hand, we lost protection from lots of diseases, and on the other hand, we have higher inflammation, and we know that inflammaging is just affecting any other aspects of aging, like in a network. So we have an amplification there. Yeah. So are you also working on the thymus or the immune system and other organs? Are you trying to rejuvenate with ERA?
Yeah, with the immune system is our next target, and we are doing this in two ways. On the one hand, we are trying to rejuvenate the T cells, meaning that the cells that are responsible for this immune monitoring function. So they also age with time, And so ERA, we are using ERA to bring them back in time and to make them, again, more youthful so that they can monitor the body and be more performant. And we have very exciting data on that. But the other thing that we're doing is, instead of working on the end product, which is the T cells, we're trying to see if ERA can actually rejuvenate the thymus per se, so that now you have a functional organ that can do the job, very well. So as you said, biology is super complex and super fascinating. You always have to shoot multiple shots on targets. And then one of the things maybe is gonna work. So
yeah, and I think you're also working on skin, right? You're working on multiple tissues, and this is just to say, the promises are for everybody. Not only isn't just about women's health. So just a question about when do you think, as a timeline, will ERA be available for humans?
it's an excellent question. I guess the primary reason for the acceleration would be obviously funding. So I do have a company, termed Biotechnology, as you mentioned that. So the company
Yes.
is actively working on implementing ERA in a clinical setup to develop products that could be used, by people, by patients. Funding is the most important, and the bottleneck. So because of course we need to generate material that can be used for human beings. It needs to be super high quality, and so it's very costly. And so that's where we are. We are very optimistic in the sense that we were hoping to start phase 1, which is the safety study, somewhere in the next year or two, again pending funding. But this kind of allows me to say a very important thing, that the most important thing to make sure first is that, forget about efficacy for a minute. We need to show that the technology is safe.
Yeah.
We don't want to create more problems than the ones that we're trying to solve. The reason why these studies are so expensive, and I understand that there's often there is frustration from people to say, oh, billions of dollars are going to research, so when are the results coming? Well, they will come, but it's a very long process, very rigorous process, very thorough process that needs to be done in the proper way to make sure that first the drugs are safe. And then once we know they are safe, we can move into the efficacy phase of the study. It's very important because, again, often people, and there is a lot of colleagues as well, I take a responsibility, indirect responsibility for that. There is a lot of colleagues actually that are, or false colleagues, that are actually trying to cut the corners, and no, that's not the way to do it. It's not the way to do it. We need to be absolutely rigorous and as scientists, and make sure that safety is the first thing to address.
Yeah. Okay, so there will be first safety trials, and probably we're looking at, how many years, 10, 20, you don't know, if you had to guess.
Well, it also depends on the program. So, for example, we are developing in the company a dermatological program, which is a little bit more at the intersection between medical and cosmetic or aesthetic. In that case, the regulatory path landscape is a little bit more forgiving and more friendly. So in that case, probably it's going to be much more accelerated. We have another program in the eye that is done in partnership with a Korean pharma that is super well established. They have a history of bringing products to phase 2 and phase 3, which are the efficacy steps. We have a very aggressive timeline there. So with the help of a pharma, I think that the ophthalmology program is going to move pretty rapidly. And we have been talking about, potentially doing something in 2 to 3 years from now. So yeah, it's definitely in the foreseeable future, and hopefully the sooner the better.
Okay, thank you very much. So in the meantime, we'll probably see something, some discoveries first coming with skin, for skin applications and eye, and then In the meantime, ladies, let's take care of our ovaries with good nutrition, avoid minimizing exposure to toxic and exercise. I will try to have an expert in women's health to tell us what we can do with lifestyle. Okay, so Vittorio, let me end. Okay, let me end with what what I call the fertility lightning round. So
Okay.
I will mention, I know this sounds like a torture. I will mention popular fertility supplement and interventions, and you will rate each of them based on a traffic light system. So is green for promising evidence worth considering, yellow for limited evidence, proceed with caution, and red, no evidence, save your money. I know you are not a nutraceutical expert,
Not.
but it's
But
nice sometimes to challenge ourselves, right, outside our comfort zone and share our opinion. These are just opinions. So I've just prepared a short list of supplements. So CoQ10 for egg and sperm quality.
okay, these I don't even know what it is, but I have no idea.
Okay, no idea. It's okay, because I think these are what people are searching on the internet, right? The idea, I think, is protecting mitochondrial health. Many of these things are working at a molecular level, at least claiming to protect based on very often animal studies. Sometimes there are some human studies that are small. Okay, so no opinion here. Inositol is a popular one. What do you think?
I mean, there is some evidence and some studies done on this. Again, it needs to be taken with a grain of salt. I would probably go with a yellow here, in the sense that most of these studies, of course, have been done in animals. It doesn't necessarily mean that it translates to humans. My take on this would be, as long as it's not harmful, why not? And probably for everything, as long as it's not harmful, why not? But I would probably say yellow.
Yeah, it's okay. 30 seconds answer are okay. Vitamin D3. Yeah.
This is a good one. The vitamin D3 does almost everything. So yes, absolutely. It's worth.
Yeah, I also think so. There are what I see is that for all the important nutrients, recommendations, especially for pregnant women, become things are where you need to vitamin D3, omega-3 fatty acids, folate. I mean, these are things that benefit everybody. But of course, when you are to think about pregnancy, you see how those things are recommended, like as if only pregnant women would need that. Now, a controversial one, NAD boosters for ovarian rejuvenation.
I would say yellow, in the sense that there is definitely some papers showing a positive effect. I haven't seen anything convincing in human studies thus far. NAD is one of the sorry, this needs to be a little longer. Sorry, the answer.
It's okay.
It's important the form you take. NAD has been shown to have an impact because it's related to sirtuins and so on. And it's the form that you utilize that is important. Some people use the NMN. So yes, there is literature, there is evidence that is convincing. Now, is NAD plus the best way to do it? I don't know. Probably the NMN is the best one. But at the same time, there is not convincing data on humans. So I'm kind of on the fence on this. Yellow.
Okay. Glutathione, so an antioxidant, our endogenous antioxidant, but injected probably or taken exogenously.
Yeah, I mean, again, it has been shown to have a plethora of positive effects. It's an antioxidant, generally speaking, is good. Does it need to be supplemented? I'm not sure. You can probably get it through a proper diet.
Yeah,
So
I also believe so. And there are other supplements that are interesting that may boost our own endogenous glutathione production. As a food, I think cruciferous vegetables do it. Actually, also through some epigenetic mechanisms. And as a supplement, there is some research on N-acetylcysteine, or NAC, especially in combination with glycine, because they are part of the pathway required for glutathione production. So it's an indirect way to support the endogenous production without taking glutathione. DHEA supplementation for ovarian function. Of course, this is an hormonal therapy, so probably with the doctor. So what do you think?
My take on this is that I think that the hormonal, but I'm not a physician, okay? So please follow the physician's instruction. And the hormonal supplementation has been suffering for a while historically. It was very popular a long time ago. Then it became super unpopular, and now it's actually coming back because now there is new formulas of the hormones that are less toxic and more efficacious.
Bioidentical, yes.
Exactly.
Yes.
So my take would be, I think based on what I'm reading is that there is more and more and more physicians actually utilizing it. And this new generation of molecules are actually way more efficacious, and they don't give side effects as the first generation did. So my take here would be green, but ask the doctor.
Great. Yes, I think that's a great take. And then DHEA supplements. So these are also a derivative of, there are compounds that are similar to those found in cruciferous vegetables, but people can take it as a pill. I don't know if you know about it and if you have any opinions.
No, I don't know much about it, but
Okay.
my take would be, and probably you definitely know better than this. Probably if you can take them through a proper diet, I think in addition to getting them, you also get all the other good things that actually that the cruciferous and other vegetables can give. So.
Thank you for saying that. I am a food first person, especially given that there are so many supplements. If we start, I have a few essential supplements that I take, but if I had to supplement everything I can get from food, first of all, there are more risks because even in these supplements, it's a matter of purity. Supplements can be contaminated. There have been cases in which even whey protein have been found to contain lead, right? You need to, everything you add, there is a risk of taking everything. So just a final question, what is the most underrated longevity supplement of practice according to you right now?
Well, I mean, let me tell you one thing. What I am doing and many other colleagues are doing is to develop next generation, interventions actually for aging associated diseases. There is already a lot we can do, and there is a lot already that we're not doing that we should be doing actually to have a long and healthy life. And I go by the obvious ones, but to me, I think that the most important one that has a solid literature now is caloric restriction, or in the sense, lowering calorie intake. I'm aware that there is not a unique, and that's another field where everybody comes with a special regimen. 1688, 2 days of starving, 1 day of fasting. There's all sorts of. But generally speaking, I think what we can all agree upon is that we need to reduce the amount of calories that we eat. Absolutely. We have to make an effort there. We have to make sure that we don't eat processed food, because it's the other most important reason why we actually age and develop diseases. And so this is, I think, the most important thing. And then all that goes with it, exercise, sleep, stress, the usual, what my grandma used to tell me when I was a kid.
Yes, going
So,
back to the roots.
right.
I agree with you on calorie restriction with a caveat. I
Yeah.
think it's probably one of the most important
Yeah.
lever for living longer for most people. But as you mentioned, also longevity is personalized. So there are people even aging like, and I always talk about my mom. She's
Yeah.
84. She's frail. She doesn't eat. I
No,
have
of
the
course.
challenge. I have the opposite
Of
challenge
course.
with her, right? To say, Mama, please eat, please,
Yeah, yeah,
especially
yeah.
protein. Yeah. So is the and this is a perfect example of it's always, it depends. We can name an effective intervention, but of course it will vary from people to people, and it doesn't work for everybody.
Absolutely. No,
The,
no, I'm saying generally speaking, of course, if you never eat, you should eat. You know.
yeah. One last question, Vittorio. What's your daily longevity routine? I know you're so busy, and we do, we challenge. I'm all, I take care of my health, but I'm not perfect. Sleep, I have lots of, because we are busy, right? But what are you trying, at least, the essential pieces you are trying to introduce in your longevity routine?
Yeah, and I mean, considering that I have a crazy life, and for example, I travel, jet lag is just killing me, so it's
Yeah.
completely unhealthy. Well, I give a lot of importance to, again, calories. I try to limit. So I eat. Well, it depends on the day, but I guess that, again, I'm not saying that you don't have to eat. I'm just saying that instead of eating, I used to eat a lot before, and you just reduce. You reduce your amount until once you are satisfied. That's it. You don't have to overeat. So that's already a good thing to do.
Yeah,
Uh,
I think,
yeah, and then
yeah. Okay,
sleep.
so
Sleep for me is
sleep.
super important. Super important. If I don't sleep at least 8 hours, I just don't function. And so sleep. And I bike to work, but I know that that's not always possible for everybody. If you don't bike, try to walk. If you can walk, if you can't walk, try to at least do something, during every day. So yeah, that's what I'm doing. I should be doing better, absolutely. But again, you have to deal with life, unfortunately, sometimes. So you have to try to use common sense. Yeah.
Thank you very much, Vittorio, for taking the time to be with us with all your busy journey. So I leave you to your very productive day, and take time to sleep tonight.
Thank you.
Bye.
Bye.
Ciao. Ciao.
Ciao.
Thank you very much. Ciao.